Swayam Prabha. Digital India. Educated India.
Good afternoon. Welcome you all to the next lecture in this particular course on analytical, spectral and microscopy applications of inorganic compounds and nanomaterials. For the past 4-5 classes we have been going through the aspects relevant to the microscopy. We have looked at the atomic force microscopy, then transmission electron microscopy, and we just began to look at the scanning electron microscopy. So, I have already mentioned to you in the scanning electron microscopy, you measure secondary electrons, you also measure backscattered electrons, and then you can use combinations of these things to generate the pictures. Though you find, of course, you can get a very good resolution in the XY plane, not much in the Z. For example, in case of transmission, you look at the electrons which passes through the matter. So therefore you are looking at the throughout the Z-axis as well. But here you see certain range of the Z-axis resolution, and thereby the image looks as if it is a kind of a three-dimensional, though it is not a completely three-dimensional, but you get a full range of the surface topology completely. And even so this way you can definitely differentiate the various kinds of species, biological systems as well. Just like I have shown in case of TEM, you can differentiate various pathogens, other things based on their images based on the TEM. Here also based on the images of the SEM you can differentiate that. You can also do, the main SEM requires a large extent of the vacuum kind of a technology, but now there are techniques which are developed even with the somewhat lower level of vacuum technology you can do. Those are called this environmental, the SEM, where you can use a somewhat less vacuum but use other conditions like the variable pressures and other kinds of things, some temperatures. You can do even a very low temperature, which is called the cryo-TEM. So these are all mainly used, and the example is shown here on the screen for this particular pathogen at a very high magnification. This is from the pollen basically. From the pollen you can see different kinds of particles, different nature of the particles, different shapes of the particles, size, shape, nature. So all of these differ. So these using a large magnifications, something like 10 times to 250 times magnification as compared to the light microscope. So we also have looked at in the previous class regarding the principle that functions in the case of the SEM microscope case. Very much similar to that of the TEM, but there are certain little differences towards the detection side. One of it is the backscattered electrons to be captured and studied, secondary electrons to be captured and studied, and any kind of you can use additional types of detectors, for example X-ray detectors, etc. So these are all kind of things. We have started also during the end of the previous class the example where the epoxy hybrids can be reinforced by using the bamboo fibers or glass fibers, particularly when these fibers are treated with the alkali. So when they are treated with the alkali, then you get much greater strength for these fibers. The reason that is explained is here as follows. As you can see, untreated bamboo fibers versus those upon treatment with the alkali. So you can see more of a random and a lot of a amorphous nature. or less crystalline nature, you can say that, more amorphous and less crystalline nature, wherein here you can see the fibrils are aligned to give a crystalline kind of a structures. So thereby, as you keep removing these amorphous things and then make more of a crystalline, and that is the reason why this the strengths of these fibers become very strong when you treat with the alkali in presence of the bamboo fibers or a glass fibers. This. Let us look at few more examples as I promised earlier. So here is one example of a porous silica and the zinc oxide. So the silica is basically a kind of a substrate. Zinc oxide is providing the nanoparticle things. So you get a composite of that, nanocomposite. So this is SEM images of porous silica, which is here as written as PSI, but don't take that as a standard synonym used, and surface before and after 100 cycles of zinc oxide. Okay, so this experiment is done with the silica, porous silica, as well as porous silica which is coated with a zinc oxide. So zinc oxide is coated by a method of atomic layer deposition, and in this case the 100 cycles of the zinc oxide has been done. So this is the N type, and this is the P type, and the corresponding zinc oxide with deposited ones. The SEM images of porous silica surface before and after 100 cycles of the zinc oxide. Okay, so here what you see is basically the silica part of it without the zinc oxide being coated, and once you come to this, you have 100 cycles of zinc oxide atomic layer deposition. So one is for the N type, the B is for the P type of that. So both are the same when you coat with these zinc oxide things. So you can see the zinc oxide composite forming, the composite being formed in this, both for the N type as well as P type. So in fact, this kind of a composite formation with the zinc oxide would enhance the properties of the light conversion properties primarily. These are the photoluminescence properties of that are basically enhanced, and this shows a wood site structure with the polycrystalline nature of that. So the produced the porous silica zinc oxide nanocomposite was tested for this for some the sensing purpose. So the sensing is done towards the mycotoxins, so aflatoxin B1 detection, so confirming the application potential. So by doing this, you can have the photocatalytic kind of activities, as well as the activities, some of the things like detection, sensing kind of activities, particularly the biomolecular systems, the mycotoxins. So these kind of toxins can be detected by using the corresponding zinc oxide layered composite of the porous silica. So as such, porous silica would not show much of those properties, but when these are made into a nanocomposite, you will have. Let's look at a couple of more examples. Something here, example which is shown for the engineering application, which means some kind of a tensile strength, these capabilities, all these aspects of it, which is referred as the failure analysis of cellulose nanocomposite. First, let us come to the figure caption, then we will look at the details of that. So this is SEM images of the fractured surfaces of samples with different carbon nanofibers. CNF is carbon nanofibers, volume fractions, and different magnifications. Okay, so A is the pure bioepoxy. This is the at 100x, and B is the same but at the 1000x. No difference in the material. It's the same, actually same sample, which is done at the 100x and the 1000x. Now this system C and D are the epoxy plus certain amount, 1% of the the carbon nanofibers. So you make a composite which is formed out of the 1% of the carbon nanofibers. And similarly here also this is 100x and this is the 1000x. We will come to the results in a while. Now the third set, E and F, again are the same, but the percent of carbon nanofibers is more, 1.3% at the 100x and the same at 1000x. So you can see a set of experiments done with the background cellulose at two different the magnifications, and cellulose plus 1% of carbon nanofibers at two different, and 1.3% of carbon nanofibers at two different magnifications. So you can see that obviously these structures are quite different here. This you have a pure bio epoxy show regular cracks indicating a typical brittle fracture kind of a system. There are no special structures, no channels, no holes, nothing like that. Now once you make this into the carbon nanofiber containing composite, okay, so you have a different kind of a fractured surface interspread with the holes and the channels, you can see. So these channels, if you look at, you enlarge and look at, you can see much better. Quite these are fractured metal as if they are like a fractured metal surfaces type. The fiber clusters have been pulled out and the resulting channels have appeared in the fractured surface. So that such fiber pull out acts as the dominant kind of a system to strengthen this composite. So because all those fibrils that you have put, you don't see it as a sticking out, but they're all there with in the surfaces, but having some interfiber interactions bring a strength into that. So such fiber pull out acts as the dominant toughening mechanism, so for the anisotropic CNF composites. So because of the weaker interfacial bonds between the CNF and bioepoxy, all SEM figures show smooth surfaces at the channels of this, and the same thing at a little higher doping or higher composite prepared with at a higher level of the carbon nanofibers. You can see that. So very nice. All those things are well spread. All those carbon nanofibers are well spread, and they form some kind of a systems where it provides a strength to this composite, and is also because the interactions between these fibers and these the polymer, the cellulose, are somewhat weaker interactions. So all these leading to a smooth channel type with which is having a tougher composite as compared to the composite without having such carbon nanofibers in that. Okay, not taken large number of examples for the simple reason because I took quite a diverse examples for case of TEM, so therefore I have avoided bringing too many in case of SEM. But I would like to bring one case here which has a set of SEMs, signature SEMs for a variety of the materials. For example, here these are all nanostructured materials imaged utilizing the ultra low the voltages are somewhat lower here. These are all called the FE-SEM kind of things. The first one, A, is for the polymer membrane used as a separator in the lithium batteries. Don't need to worry about this potential at which measured, etc. Not important in this for us in this case. So this is for a polymer membrane, you can see that, which is used in the lithium batteries as a separator. And the B is the lithium battery positive electrode material. So this is used for the positive electrodes. So this is for the membrane as a separator. This is for the positive electrode material. And the C is a cross section of polymer sheet with graphene flakes. You can see that this graphene sheet with the polymer and nanoparticles. And this is the image. So entire thing set up covered to that. D is a nanoporous oxide particles. So the oxide ones. and E is anopore inorganic membrane. So this is for the aluminum oxide, which is used for the filtration. Okay, and the F is blue toner particles imaged at this. So what I wanted to convey to you is, at this stage, it is not that to remember any of these things. Materials having different nanostructures, microstructures would be reflected in their SEM pictures accordingly. So you can study their pore size, you can study their topology, surface structures, and the shapes. Also you can make the geometries of these. So many of these things. You can also identify some of these chemical species, not shown in this particular set of experiments, but you can do other things as well, like identifying what kind of a distribution of the species, atoms, or some other things which are present in that particular kind of species. So that is where this statement will tell you the SEM provides unparalleled flexibility through the addition of an assortment of electrical, mechanical, and chemical test equipment, making the instrument a self-contained nano laboratory. So SEM itself, you're equating it to a nano laboratory. So you can study the samples for their mechanical aspects, which mechanical engineers use, chemical aspects and electrical aspects of all, where all materials used in all these contexts are this. So this article provides a development of SEM technology that enable to see the new nanostructured materials with diverse applications. And the corresponding reference is given here. You can see it is a review article. It focuses a variety of techniques, variety of materials, how to get their optimal kind of a pictures in that. Okay, so one another example that I have brought in this particular topic is that the hybrids can also be synthesized. So instead of composite, we can call it as a hybrid here. So this hybrid is the inorganic part and there is a protein part. The organic part is the protein and the inorganic part is the inorganic material. In this example, it is copper phosphate. Not always inorganic means copper phosphate. That should not be taken. So the copper phosphate and the organic part is the protein. This protein is Moringa oleifera coagulant protein, okay, which is basically isolated from drumstick seeds, and this is being done. Isolate this protein and you try to coat the copper phosphate system with these protein molecules. That's where the thing is. Now, here with respect to the SEM, I would like to demonstrate the following. So, inorganic protein hybrid nanoflowers, because the structures look like the flower shape and they are at the nanoscale. So, that's where and that gives a very good surface structure, surface availability for reactions, and they are good catalysts and they have good porosity as well, and a large amount of surface area. A large amount of surface area is required for materials to act as a catalyst, very well. So nanoflower growth dependency and the concentration and time as studied by SEM. So this is a system which is done between the copper phosphate solution in some buffer and along with the Moringa oleifera coagulant protein added at different concentrations. And so here no protein at all is there, and you can see the kinds of things. Some sheets are formed, but there is no flower at all, one can imagine here. And you add this protein to an extent of 0.025 millimolar into the second experiment. So in that you can see. Now you can start seeing there is some shape coming up which looks like a flowerish kind of thing. Now you take another experiment where you have added much more of the protein, 0.1 millimolar of the protein, as you can see. It's a much better and quite well defined petals are formed, and the petal strength and the density seem to be increasing from 0.05 to 0.1. Now you go from 0.1 to 0.5 millimolar of protein, then in that solution you get a very dense, very packed, highly packed kind of things. So what you see as you go from the left to the right with no protein almost no flower kind of a structure. With a protein small amount, some flower starts making, and with a little higher, somewhat well defined with more petals, and at a higher concentration, the dense petals, and one can fully see the fully grown nanoflower. And here you can see nanoflower size. If you put as a function of the protein concentration, you can see it's growing like this. Okay. Also, as we have seen, the density per given unit area, the number of petals, and you take a large number of examples and try to count and then plot, and that is what is done here. And those, if you look at that here, the this is the petal density. Petal density is, as you increase that NF size, nanoflower size is increasing, nanoflower density, this is density. Here you have to take this particular scale. Okay, so at the lower and higher, no, it is the reverse. There is some little kind of a thing. Nanoflower size 10, 20, 30, 40 micrometer, and the petal density, as you increase, the petal density is basically increasing. So it is a reverse plot, and this, the labeling, the numbers are given wrong. So you need to look at these numbers as a reverse of these ones. So the relative petal density is absolutely increasing. Of course, nanoflower size is also increasing in that. Okay, so, and the second thing is, suppose you start this reaction and at certain level, there is 0.1 millimolar of the protein, no change in the protein concentration, but only time. So as you increase that, immediately almost nothing, and after 30 minutes you can start seeing some, and after the 66 hours you can see more flower and many more flowers of that. So these are all quite well established by SEM. Okay, so you can establish. So all these are being explained here, and in this particular paper you can find. So elemental mapping, if you take one of these flower and try to look at the elemental mapping, as I told you, these SEM machines are also equipped with elemental mapping kind of thing. So each one of the color correspond to one other of these carbon, oxygen, phosphorus and copper have been mapped here, and that shows that there is a quite well distributed of all these elements in this flower to ensure that this is made up of this coming from copper and the phosphate, and the protein will have again the same, the oxygen, nitrogen, carbon and the phosphorus very little extent. So you will have those things. So those are all coming into this kind of the mapping structure. So the mapping is used to identify the chemical composition, distribution of the chemical entities, etc. in these ones. Okay, like in any other topic that I also bring in this particular the exercise in order for you to enhance your practice, enhance your understanding by practice, by extended practice. So we have basically covered in this the three techniques. One is the AFM based things, where we have looked at the large number of examples based on the AFM. We have also looked at TEM and the SEM. So AFM examples are here. You can see the structures of the plasmids, and this is the nano composite which is formed from graphene oxide into the ethylene methyl acrylate kind of thing. Then we looked at the silver nanoparticles into the chitosan, and then we have looked at the epoxy nanocomposite with the exfoliated graphite or carbon nanofibers, and these are all used to look at the strength of this composite later on, and that's where the thing is that. And we have also looked at some engineering applications of these by the nano rubber, which is being impregnated with the multi-wall carbon nanotubes at different concentrations, and then based on that we have looked. And the regions of the Young's modulus and the regions of the adhesion maps, everything is being developed in that. So then we have looked at the TEM, and we looked at the huge number of example domain. For example, nanoparticles of the gold nanoparticles of the different shapes, different sizes, their impact on the cell properties, etc. All of these we have looked at, how well the cell survival takes place, and then we have looked at some nanocomposites, graphene based nanocomposite, and the mechanical properties. Of course, we have not talked about the mechanical properties, but these are made with the idea of making that. And we also looked at the graphene oxide based magnetic nanocomposites like cobalt, CoFe2O4, and their crystal structure, their domains are being identified. And you can also look at these silica based composite, which forms different aggregates and the overall aggregate individually, using both the TEM pictures being modeled by using the small angle X-ray scattering. And then we have also shown that electron energy loss spectroscopy at the near edge can be used to identify the kind of a coordination spheres that you have, and this can differentiate the materials present in the superconductor, non-superconductor, and explain. You can also quantify the percentage of different kind of coordination spheres present in these. You can also map different elements present in these things, but whether they are all kinds of things you can map, and that will give you regions where the different kinds of the elements are being present, and based on that the properties, engineering properties later on that you're going to measure can be correlated with that kind of thing. You can also measure the structures of the protein, structures of the virus, nature of the virus, particles, etc. And just in this class I talked to you some examples of nanocomposites and nanoflowers. So I think in order to keep in pace with the course, you need to do a certain level of practice, make these things a more familiar, understandable, and knowledge acquisition by the candidate. In the lectures I have provided the AMF application towards the mechanical properties, but this AFM can be used for the conductive force, conductivity, and electric properties like redox potentials, etc. So I would like to recommend you to get practice, that you should pick up some examples using the AFM data. One, in case of the conductivity measurements, and discuss its plots in interpreting the regions responsible for the conductance. So there are regions, there will be plots which shows you the region where, just like here we showed in this example for this their tensile strengths, Young's modulus, these kind of things we have shown, and then also their adhesive forces regions. So all you can make the contours plots. So those kinds of plots, contours, you should try to interpret and try to correlate with the material surface structure with the properties of that. Similar exercise as in one, using the AFM data with the electrochemical potentials, redox. Interpret the nanometric regions responsible for the redox process. So there you can expect for the conducting regions, here you can expect for the redox kind of regions. So what, of course, they are related. Okay, so that is for the AFM. You can do do a large other kinds of things also from the literature. A lot of data is available. In order to acquire practice, pick up TEM data for two catalytic processes where heterogeneous inorganic catalysts are being used, and compare their TEM obtained before the catalysis and after the catalysis, and provide your conclusions regarding how TEM is able to support the functioning of the catalyst. So this is very important, and you can do for a few cases of that. If the data is available after several cycles, then the similar exercise will be compared and addressed regarding the functioning and how this will be. Similar kind of an exercise that we talked about under TEM, you can also look at with using the SEM. More importantly, in these things, there's a lot of data is available with this EDAX data, ELS data, and all these kinds of things, and elemental mapping, both in TEM as well as in SEM. So you should practice some of those, and so that you will be able to see the regions where these materials or these entities or these atoms or these species are distributed, and how the property correlation will be helped by this kind of things. So I think these are certain things which would cover within these number of lectures, starting from the analytical, then spectral, the diffraction, to the microscopy techniques of this. We will try to spend one lecture to look at all these, what all we have done in one last lecture. We will look at those kind of things, what all I have focused, and how one should be sort of taking up this, where would this lead to, etc. These kind of aspects can be discussed in one last lecture of that. Thank you very much.